problem, the research, hypothesis, the experiment, how we set it up, the variables. Our last couple things are the observations. So observation is, this is when you're collecting your data. You're gathering the results of your experiment. And sometimes you do this once at the end of your experiment. Other times you may be doing it several times as the experiment is progressing. It depends on your experimental design. Okay? And remember, there are two types of observations: quantitative and qualitative. Quantitative, which are numerical, and qualitative, which are not. Okay? We use data tables, we use charts, we use graphs to portray our data and to show it in different ways. And then once we have that data, we can analyze it. We look at the data and say, okay, based on these results of my experiment, was my hypothesis correct or was it incorrect? We make a decision. I talked about this and I kind of rushed at the end of class. So significance is something important. You know, if we collect our data and there is a small difference between our control and experimental groups, well then you have to make the decision, well, is that difference enough to be what we call significant? So, you know, if you do this fertilizer experiment, you grow two plants, one with fertilizer, one without. And the fertilizer plants turn out to be 17.3 centimeters tall after your time period. And the non-fertilizer is 17.2 centimeters tall. Well, did the fertilizer work? Fertilizer plants are 17.3. The regular plants are 17.2. The average of the fertilized plants is greater than the non-fertilized plants, right?
So it worked.
I don't know, did it? Did you say it did?
Not a substantial.
So if you just said it's not a substantial amount.
So it could be a random occurrence.
Right. So we have statistical means. We're not really going to get into them, but you will in high school probably. That there are certain statistical methods we can use that you can tell, okay, if there is a difference, is it enough to be what we call significant? Is there a significant difference? And what you do is you say, okay, I want to be 95% confident that this difference is due to my treatment. And doing some statistics, you can look at the difference and say, yes, this is a significant difference. Or you can say, no, it's not. Probably that case, 0.1 centimeters, is probably not a significant difference. We can't really say that the treatment had any effect. That could most likely be due to just random chance and not necessarily our treatment. So you have some statistics you can do that help you make that decision. All right, so once we are at our conclusion step, we will either determine our hypothesis was accepted or rejected. If it's accepted, well, we probably could repeat the experiment again to confirm our results. We can be sure of our experimental design, to be sure it's a valid experiment. And then often what scientists do is then this process called peer review. In peer review, basically you write up a report of your experiment that you did. You give an overview, you tell your methods, tell how you collected your data, you show a summary of the data, you determine whether your hypothesis is true or not, you analyze and tell what this means. And then it's submitted usually to what's called a peer-reviewed journal. Okay? If scientists subscribe to these journals, you know, if your specialty is in, you know, the caterpillars of the deciduous forest in the Northeast, there's probably some journal that you subscribe to and you're a member of the society, and every month you get in the mail a little magazine-like thing that has several articles describing other scientists' research on caterpillars in the northeast deciduous forest. And that's how scientists share. And they can look at the experiment and say, oh, okay. Or maybe I don't think that was a good experiment. I think there's this problem. And so they review each other's work to sort of double check it and to share it with each other. And there are thousands and thousands of these peer-reviewed journals. They're very important in medicine. Doctors are always reading. Their specialty is in cardiology. They're probably reading cardiology journals where other doctors are publishing research about various treatments and how they help. And that's how they learn about new treatments and how they work and understand those things. So scientists share their research using these peer-reviewed journals. Then they can ask a new question, start the process. Other times, our hypothesis may be rejected, and that's okay. And rejecting your hypothesis still gives you valuable information. It can still lead to a new investigation, new ideas, new avenues of research. And then you could create a new experiment and repeat again. So the question we have to answer is, well, what makes a valid experiment? And we use this word all the time, valid, validity. It sort of means reliable, a trustworthy experiment where we can be confident of the results. And one important part is the sample size. Okay? A larger sample size makes for a more valid experiment. It reduces, like Antonio was saying before, outliers. These data points that for some odd reason don't seem to fit with our data, and they could just be due to random chance. Lots of data helps average out those outliers. The more samples we have, we can have greater confidence in the results. A good experiment has a single independent variable. Now, oftentimes that's impossible. You really can't, if you're doing a medical study or something, comparing two treatments and two groups of people, well, you can't have the same people, okay? But you try to carefully control as many external factors as possible so that there's only one main independent variable. And a valid experiment is often repeated several times in order to confirm the results. If you do an experiment again and you come up with the same results, you can be more confident in those results.
Who wants the video?
In a minute. All right, so we're going to look at two examples of the experimental scientific method, setting up an experiment. So let's start with this one. An experiment with 30 pea plants, 10 given no fertilizer. 10 are given 5 grams of fertilizer. 10 plants are given 15 grams of fertilizer. After 14 days, the height of the plants is measured. Okay, let's fill this in. What hypothesis is this experiment testing? The experiment is done to test a hypothesis. What hypothesis is being tested here? Sophia?
If an increased amount of fertilizer will help the plants grow taller.
Yeah, that would be one possible hypothesis. And if I phrase that a little differently, if pea plants are given additional fertilizer, then they will grow taller. Remember, we often want to state our hypothesis as an if-then statement. If I change the independent variable, then the dependent variable will change in this way. Okay, so if we're going to set up this experiment, what is our independent variable? Caitlin?
The fertilizer?
Let's just be a little bit more specific. Because it varies brands of fertilizer.
On the same brand?
Yeah, but so what is different about it?
The amount.
The amount of fertilizer. Yeah, the amount of fertilizer is what's going to be different. It's what's different between my groups. My 3 groups is the amount of fertilizer. And here's where it's important to be specific. Because Caitlin said just saying fertilizer could mean different things. Maybe I was testing different brands of fertilizer. Okay? Or maybe the same type of fertilizer but different forms. Okay. What would the dependent variable be in this experiment? Isn't it?
The height of the people.
Yeah, the data we're collecting is the height of the pea plant. Again, we want to be specific, not just the pea plant. And we also don't want to restate the question. We don't want to say which plant grew tallest. Okay, just tell me, what are we measuring? We're measuring the height of the pea plant. How about some variables that would have to be controlled? Variables that would have to be held constant. Give me one, Nate.
The amount of water given.
The amount of water that they're given. Matt?
The brand of fertilizer.
The brand of fertilizer.
The type
Sophia?
of people.
The type of pea plant. Correct. All those things. We can make a wrong with this, but. In this experiment, what's the control group? Grace?
The 10 plants that are given no fertilizer.
Yeah, the plant's given no fertilizer. Control group is the group we need as a comparison. Group treated normally. And then the experimental group. Mosquito.
Plants given fertilizer.
Well, yeah, the one's getting the different amounts of fertilizer. We can have multiple experimental groups. How can I make this a better experiment? How can I improve on its validity?
Get more plants.
Use more plants, a greater sample size. 20 in each group would be better. 50 would be even better.
What is this about?
All right, so any questions about scientific method? We're going to just watch a little documentary about scientific method in action.